Hi guys, it's another biology with Miss Cherry. We've been doing this for about 7 million years. I think it's only been about 4 weeks. But here we are. This is lesson 12. We're looking at evolution today. So, what is evolution? What do the graphs below show? So you've got the number of individuals up the y-axis and the phenotypes of snails, I guess, on the x. And how would you define each selection type? And we're going to take a look at those in turn over the lesson today. But hopefully this should get you thinking, to begin with, about the differences between them. So pause, spend a couple of minutes on these, and play again when you're ready to go. So as I've said, this lesson is entirely linked to evolution and selection, and it links very heavily back to Module 4, so looking at natural selection, the types of natural selection that can occur, normal distribution, and things like that. So we're going to be having a look at each of those different types of selection and the selection pressures that might cause them, to explain the effects of each of those selection pressures on evolution or the rate of evolution, and then to apply our knowledge, to apply the types of selection and that new gained information to the peppered moth case study. There's also another case study that we're going to have a look at about the lazuli birds, but I'll cover that when we get to it. So let's get started. First thing I'd like you to do for me is define evolution. So write your own definition exactly as you would do in an exam. What does it mean? And then maybe give me some reasons as to how it happens. Pause, play again when you're ready. Alright, so we'll start with the definition then. Evolution is a change in inherited characteristics of a group of organisms over time caused by changes in allelic frequency or allele frequency within a population. Notice that we're talking about characteristics that can be inherited, because remember there's those environmental influences as well. But for this example, for this lesson, we're only interested in the genetic side of things. Now it can be affected by a few different factors. So it can be helped by mutations, selection, so sexual selection of a mate, and natural selection, so the selection pressures on a population. But also, immigration and emigration have a significant impact on the gene pool, so people or organisms coming into or leaving an ecosystem or environment. Okay, so even if you think about the migration of the human species, we all originated from Homo sapiens in Africa, and the migration of those early people, so the emigration from Africa, mid-Africa, out towards Europe, is going to then affect the gene pool and develop the Homo sapiens species in those areas where they weren't originally. So we've spoken about the genetics and things like that, and this whole topic relates around the idea of population genetics, or how allele frequencies in a population change over time. And we know, as I said with the Homo sapiens example, that studying those genetics help give us evidence for evolution and evidence of the migratory patterns of certain organisms. So in larger populations, we assume that there's going to be lots of genetic diversity within them because of that vast gene pool. And that's going to give a huge amount of variation. If you think about Homo sapiens, for example, humans, modern day humans, there's so much diversity. We've got eye colour, hair colour, skin colours, skin textures, hair textures. There's loads of stuff that's different between all of us. There's also a lot that's the same, but there's a vast amount of genetic differences. And the populations are able to either adapt to selection pressures, so respond to the environmental changes, or, if they can't respond to environmental changes, allow natural selection to take place to start influencing the process of evolution. Okay, so if you can't adapt, if you can't survive, if you can't reproduce, then some of you will die. Which scientifically is a bit rubbish if you're one of the poorly adapted ones, but it is what it is. And we're going to take a look at the 3 types of selection today. And evolutionary selection is as defined in that green sentence, when changes in evolutionary pressures affect the most advantageous phenotype in a population in an ecosystem. So we're looking at a specific group in a specific point of time. And we're seeing how the most advantageous phenotype is changed. So how is the distribution of that organism, that phenotype, changed? Got 3 different types we're looking at: stabilising, directional and disruptive. Each of them has a graph to go with them, and each of them has got a key example that you might want to take notes Unfortunately, for this first bit, it's a lot of me talking, so I apologise in advance. I've been talking for about 3 hours by the time I do this video, so yeah, if my voice goes a little bit ropey, I'm sorry. But, bear with me, we'll be fine. So the first one we're going to look at is stabilising selection. And I want you to think about how the birth weight of babies is a really good example of natural selection. You can see at the bottom we've got some cute little bubbas, not very genetically diverse I'm afraid, I apologise, but you can see already that there's some that are a little bit chubbier, there are some that are a little bit thinner, there are some that are a little bit shorter, some that are a little bit taller. How is the variation in size, especially at birth, an example of natural selection? Now hopefully you've had some time to think, you've got some decent ideas, although there's also a high chance that you've just left this video running so you're on here now. But the first thing we need to recognise is that average birth weight babies are going to be the most common. Okay, most of us were probably born maybe around 7 pounds-ish, generally a standard average weight. If your baby is slightly lighter, so towards the lower end of a weight range, there's chances that they might be premature, so they might be underdeveloped, but also in that underdeveloped state, maybe they've got a weaker immune system and therefore they're more prone to infection, which means, less likely to survive long term. Conversely, at the upper end of the scale, so again, towards the higher end of weights, but in our normal distribution curve still, the higher the birth weight, the more difficult birth is going to be. So maybe you might get stuck in the birth canal. Maybe your mum has to have a caesarean section. Maybe you get a little bit stressed during the birthing process. And overall, it's going to be slightly more challenging to deliver you. And therefore, again, there's a chance that you are less likely to survive. Now, both of those extremes, the low and the higher birth weights, reduce the chance of survival of the babies, and also in some cases the mum, but we're only focusing on the babies for now, and therefore the more average weight babies survive. And if those average weight babies survive, they can reproduce, and when they reproduce, it's likely that because of that alleles being passed on, that baby that they have is going to be of an average weight. And if we look at it on a graph, it looks like a very, very narrow normal distribution. So you can see in the picture, we've got a dashed curve, and that's our broad original population, our normal distribution. But over time, if you imagine that the babies with birth weights down there and up there are going to maybe die or not survive, or maybe not reproduce, then the range of birth weights tends to narrow up. Okay, so we say that stabilising selection eliminates extremes in the phenotype. Okay, and it reduces the capacity of the population for evolution. And that's because we're going to get a narrowing of the gene pool. We're going to reduce the likelihood of heavy baby alleles and light baby alleles in the population. So they're not likely to be as passed on. It means that the average baby is the most common. They're generally just standard what average means. But this is what happens over time. Okay? And this is what happens when a population or an environment is stable. So there's not a lot of change in the environment. Not a lot of change in environment. So an example of this might be, again, humans. You could pick another organism if you really fancied. I find humans quite easy to use because we're living it. But our environment is generally quite stable. There's enough food for us if we're lucky enough or privileged enough to have enough money. We've got houses, we've got roofs over our head. It means that our sphere of change is very, very small. And therefore, there's no real need for loads of really heavy babies. There's no real need for lots of light babies. That's not to say that they're not valued as individuals, but evolutionarily speaking, there's no benefit to having either of them. An example down at the bottom we've got, if temperatures remain at about 10 degrees Celsius, animals with medium coats will be able to survive small fluctuations in temperature better. So if we're thinking about Arctic ecosystems maybe, or maybe we're thinking about mountainous regions. If an organism has got too long a coat, maybe they've evolved or mutated to have a really long woolly coat, they're going to get a little bit too hot, and then we get effects on things like enzymes. And likewise, if their coat is too short, then they're going to get too cold and maybe they just perish, maybe they just die. The second type of selection is directional selection. And remember, we're talking about the effect on the most advantageous phenotype. And directional selection is where the most common phenotype isn't any longer the most advantageous. Okay, and this would be generally caused by a change in environment. And more specifically, a change in selection pressure. And the example we can see here is, or in the diagram, is one that is most often dragged up with this topic, especially in paper 3. So in this directional selection, we end up with the less common or more extreme, so either the top end or the lower end, phenotypes being selected for. And we say that they are positively selected for. And this directional selection is really the start of evolution of a species. Now, the key words that we need to be able to use to describe this change is that the allelic frequency shifts towards the extremes or the extreme phenotypes. And ultimately, this is going to result in a change of the gene pool or the combination of alleles in a population. Now, in the diagram, I'm not going to ask you this question. We would discuss it if my animations would actually work. But bacterial resistance to antibiotics is the example for this. Okay, so we can see in the diagram that an initial bacterial population showed by the dashed line has a low drug resistance. However, over time, the majority of the population actually shift to having that drug resistance. So on the next slide, there's going to be some blanks. I'd like you to pause the video and fill them out for me using your knowledge about bacterial development and evolution. So here you go. Have a go at this. There's 1, 2, 3, 4, 5 sentences and quite a few gaps. If you're not too sure, have a guess anyway, and you can amend your answer on the next slide. So pause. This should take you maybe a few minutes and then play again in a sec. So the first sentence, bacteria show genetic variation and have a large gene pool. And some bacteria have alleles for antibiotic resistance. They develop that mutation. Now, normally, the alleles don't have an effect, but when antibiotics are added to a growth medium and the selection pressure, so those antibiotics are now a selection pressure, changes, those mutated resistance alleles become advantageous. Bacteria without the resistance are selected against, while bacteria with the alleles are selected for. Okay, so we're positively evolving to have lots of population with that mutation that gives them resistance. And over time, the allelic frequency changes, shifting the mean, so the mean allelic frequency, the most common phenotype, towards the more extreme phenotype. And generally we shift it upwards, especially in this case, towards the resistance end. And the final type of selection is disruptive. And this is really quite uncommon in a species, but it's quite important in evolution. So when is there an example? You're welcome to think about it in terms of cats. I love cats, that's why I've got loads of pictures of them, but you might also want to choose some other organisms that show a lot of variation. When do you think the extremes of a phenotype might be selected for at the expense of the intermediate phenotypes? So when might the two cats at the far ends of furness, so either no fur or lots of fur, be more advantageous than this middle moggy? Pause and play again when you're ready. Disruptive selection occurs most when an environmental pressure takes at least two forms. So it might be when you vary from really hot to really cold. Cats are a great example of this because the UK does see quite significant fluctuations in temperature. I've got a cat that's very similar to the far right picture. She's a Maine Coon. She's quite fluffy. And in summer, she's a heap of rubbish. She's useless. She just lays there stretched out to try and cool down. But in the winter, she's chill. She loves it. She's really fluffy, so she gets nice and hot. Now, which cat is best suited to the cold? You might want to think about what I've just said. And which one is best suited to the hot? And what's going to happen to these middly phenotypes, the ones that look like just that standard moggy cat in the middle? So disruptive selection means that the two extreme phenotypes are selected for. So in a hot condition, the short-haired animals, maybe these horrible little sphinx cats down on the left-hand side, will be selected for because they're less insulated, they can lose body heat a little bit better. Whereas in cold conditions, cats like this Norwegian forest cat are fluffier, they can insulate themselves a little bit more. But these boring average cats that most people have isn't really useful for either. And therefore, in significant situations, so maybe if all cats lived outdoors, these average hair length cats are going to be selected against. And thinking about that, where the average phenotype is selected against, what do you think the graph would look like for disruptive selection? Have a little sketch, and I'm going to show you in 3, 2, 1. So the graph actually has a dip in it. Okay, it's got two peaks where the extreme phenotypes are, so at the lower end where you've got less fur, and the upper end where there's lots of fur. But actually, the most, or what was previously the most common phenotype, is no longer the most common. In fact, it's the lowest phenotype. There's no point in organisms having those. Now, some organisms exhibit disruptive selection behaviours, so in terms of colouring, for example. So I'd like you to read this scientific article. You can just read the introduction if you like, or you can just read the abstract, the very short summary at the top of the page, of Green et al.'s paper, Disruptive Sexual Selection for Plumage Coloration in a Passerine Bird. So they're looking at the lazuli bird in here. There's a lazuli bird in the picture. You can see it's quite jazzy looking. I just want you to tell me what they found out in this study. It's not an exam question, it's just getting used to reading scientific papers. This point I really need you to flag up, I really need you to have your attention drawn to this. This selection process, the types of selection, do not happen from mother to child. They're not going to happen even from grandparents to child. They take generations after generations after generations to appear. It's a really long process. Little changes can happen, but the whole idea of producing a genetically distinct species, that evolution and process as a whole, is slow. Please, 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 in these exam questions, do not make the mistake of assuming that it's quick and it's going to happen from your mum to you, for example. No. Straight up, no. Not going to happen. Now, it's your guys' turn to do a little bit more independent work, so I've put a little photo on the OneNote pages. It's Selection in the Peppered Moth off of page 194 in the Oxford book, if you've got your own copy. Oh, wrong pen. O-U-P. Want you to answer those questions for me. What type of selection selection does the peppered moth show? What evidence is there that the peppered moth show this type of selection? What is the selection pressure? And then sketch what you think the graph would show. Think maybe back to the starter where we had the range of snail colours. And show me the example or the distribution of phenotypes before and then the distribution of phenotypes after on that curve shape. And then, again, like every other time that I've had homework on a slide, forget about it because you're still at home. Have a go at the summary questions on page 195 and question 4 from page 201 that I've also taken a photo of, and it's on OneNote for you. And I will give you the answers as well at the bottom of the page so you can self-assess and see how you've done. As per usual, we're going to finish off a lesson with some exam questions. So there's only a couple of them this time, and we'll go through the marks individually at the end. The graphs below show 3 types of natural selection. The shaded areas marked with arrows show the individuals in the population which are being selected against. The dotted vertical lines show the statistical means. What names are given to the types of selections A, B and C? Should be quite simple. A, B and C are what different types of selection? Pause, play again when you're ready. Now for this next question, you need to draw the graphs on the 3 sets of axes below, you can sketch them in your notes, to show the distribution in the phenotypes of A, B and C after the selection has operated for several generations. And you need to indicate the new means with dotted lines. So there's 6 marks available here, 1 for the curves and 1 for the means. So think back, maybe look back or scroll back to bits of the video where we did those changed graphs and have a go, and we'll go through the answers again in a second. Now the final question, describe one specific example of the type of selection shown in graph B, which looks or looked a bit, and I'll do the little shaded bit in here, naming the organism and describing the character selected in your answer. So if the shaded bit is removed, what types of selection is that? And give me the organism and describing the character selected in the answer. Okay, the next slide shows the answers, so play again when you are ready. So for question 1, we have got a stabilising selection, directional selection for B, and disruptive selection for C. And graph-wise, they should have looked like this. So if we remove the stabilising selection extremes, you get a really narrow graph. If we move the directional selection bits up, then the mean shifts, but also the entire graph shifts up. And finally, for that disruptive selection, we've got 2 means. So you won't get the mark if you haven't got those 2 mean dashed lines there as well. Now the final example is that we're looking at that directional selection. So you could have gone for bacterial resistance, that's fine. And you would have said, describe one specific example. You might say bacterial resistance in bacteria, and they are selecting for antibiotic resistance, and that's going to increase over time. The exam paper, for some reason, told me about artificial selection. So you could have given an example of an animal, maybe some sheep, looking to improve the wool yield. I don't know. You could have picked some of those if you fancied it. If you didn't get it right, though, maybe it's worth making a few notes about those in the meantime, in case a question about pigs ever comes up. Alright guys, that's the end of the lesson. I hope you found it useful. Let me know if you've got any questions, and I will see you later. Bye bye.